real pleasure for me to be here and speak to this audience because I think I live in this world that between the clinical and the environmental science, and it's interesting to navigate that because the audiences, they do not have necessarily from each other understanding or experiences in the different worlds, and I try to see how we can link this and we can to create teams of scientists who are clinicians and, for example, environmental scientists can work together. And I think more and more we see the relevance of environmental exposures and health. And so today's talk is going to be about translational research in environmental science, understanding mechanisms of airway infection. The reason why I put that is because I want to give you an example of where I tried to do some translational science in regards to the environment. So some of the disclosures, I'm a clinical director for the Integrative Health Science Facility at the NIHS that is supported here at the HSRC. Director of the Clinical Research Unit, which is the Institute of Clinical Translational Science for the university. And I have funding from different NIH sources. And, you know, I also want to invite anybody who has any questions in the middle of the talk to feel free to ask or any comments. That doesn't need to be that formal. We want to make it very dynamic and interactive. So the first thing I want to do is come from the world of the clinical aspect. And I want to give you the context because all these about editorial back in 1974 in the England Journal talking about bench to bedside. And that's how this whole conversation began. And then more recently, we know that the NIHS has put a paper in 2018 about translational science. A commentary actually came out in 2019 that how you take some of the paradigm and the framework of translational science in the clinical aspect to environmental science. And we do not have the answers yet, but we have some examples. We need to work more towards that. So the NIH, when you think about translational science, the ultimate place is the clinical trial. That's the center where we think about this. But there are other aspects. But the NIH definition of a clinical trial, they say, is a research study in which one, could be just one, or more human subjects are prospectively assigned to one or more interventions to evaluate the effect of those interventions on health-related biomedical or behavioral outcomes. So, for example, we have an exposure chamber, and if you put somebody in that chamber and you expose them to humidity or temperature or some diesel exhaust, that is an intervention. That becomes automatically a clinical trial. Even though we're not doing any drugs, we're not doing absolutely anything. Having an intervention on these humans, it becomes a clinical trial. A research study in which one or more human subjects are prospectively assigned, this term prospectively assigned refers to a predefined process. Either you randomize, specified in a approved protocol that stipulates the assignment of research subjects, and you can have intervention, a placebo, or other control, and it's part of a clinical trial. So that would be the assignment that you're given to this subject. And I use the word subject because that's usually the way we talked about clinical research, not patients. When you talk about patients, it's going to be an research project. We mentioned that these are subjects. This is to one or more interventions. So an intervention is defined as a manipulation of the subject or subject's environment for the purpose of modifying one or more health-related biomedical or behavioral processes. So it could be drugs, it could be devices, it could be strategies to change health-related behaviors, it could be treatment strategies, prevention strategies, and even diagnostic strategies, which may include even placebo or controls. All that becomes part of that intervention. If I put a Fitbit on somebody and I want to see how much exercise will increase, the information of the Fitbit on that subject, knowing that potentially changing their behavior because they're using the Fitbit. And is in the concept, you evaluate the effect of those interventions on health-related biomedical or behavioral outcomes. And what I mentioned to you right now is I don't exercise. I put now an instrument that is going to measure how much you walk, and you start walking more. That's a behavioral outcome. So when you look at, in the NIH website, when you look at the definition, you use the following 4 questions to determine the difference between a clinical study and a clinical trial. And I'll tell you a little bit more about that after this slide. So the questions are, does a study involve a human participant? Are the participants prospectively assigned to an intervention? Is the study designed to evaluate the effect of the intervention on the participant? Is the effect being evaluated a health-related biomedical or behavioral outcome? So know that if the answers to the 4 questions are yes, your study meets the NIH definition of a clinical trial. Even if you're studying healthy participants, your study does not have a comparison group, your study is only designed to assess pharmacokinetics, safety, and a maximum tolerated dose of an investigational drug, or your study is utilizing a behavioral intervention. So if you have yes to all those 4 questions, even though you're not thinking in the typical aspect of somebody with disease, that's already a clinical trial. More recently, the NIH has come up with another funding process, which is called BESH, which stands for Basic Experimental Subject, BES, basic experimental studies in human subjects. And that is really trying to use some of these basic mechanisms that you're trying to understand, for example, respond to carbon dioxide. And it's only looking at mechanism in the context of that you can really do in animal model, for example. That mechanism, it comes through requests for applications or certain RFAs that the NIH comes out, that they say that this is a BESH study. It's really about basic mechanisms. And the advantage of the study is that it becomes really a basic science type of research that does not require a registration of your study into clinicaltrials.gov. So clinicaltrials.gov, it is very cumbersome process in which you need to register your study and you need to make sure that you're reporting what are the outcomes of that study. So the moment you have a clinical trial based on these questions answering yes, now you have another layer of regulations and also processes that you need to include. So the bottom line is and research without all the burden of a clinical trial. So the phases of translational research, and this is an important slide that I'll try to put in the context of environmental science, are 4 components. A phase 1 translation that we're going to call it T1. So research that seeks to move a basic discovery into the realm of human health. So that's a typical scenario that you think about the bench and understanding what is the bench to explain something that you have observed in a patient. A T2 or phase 2 is a research that assesses the value of early T1 research to develop evidence-based guidelines and the best practices. So here really what you're trying to do in a phase 2 is whatever you learn from that this intervention, this mechanism actually alters the disease. You have a drug that seems to work, and now you're trying to put that into the into the clinical practice. And in a phase 4, T4, research seeks to evaluate the real world. So we call this basically the drug has been FDA approved, now it's being used, but instead of these very rigid inclusion exclusion criteria that patients had or subjects had to go in to get enrolled into the study, now every practitioner is using this drug out in the community. You're not necessarily taking into consideration all these exclusion criteria. You're giving this inhaler to people who has asthma, but they are smokers. And in the clinical trial, the person who were smokers and had asthma was not able to get enrolled. So now you start to understand how it's working in the real world. So these phases, they can overlap, and there's a feedback on one another to facilitate the integration of new knowledge. So what do we need to foster translational research? Because in practice, translation is difficult to achieve, in part due to research training, and there is compartmentalization. So basic scientists are not trained to think of their work's clinical application. Clinicians are often not taught to develop research questions based on clinical observations, and the public health scientists have an understanding of the impact of the changing knowledge on the community, but they may not have a strong background in basic and clinical research. And what is happening is that we have all understood this in the past, and now there's a clear articulation of what we call team science. And team science basically is bringing people from different walks of life, different expertise, and trying to tackle complex problems. And when you think about translational research is to be tackling complex problems. Okay. So, translational research in environmental science. So we talked about the T1. And I'd recommend you to look at this article. It's really well written by Kaufman and Curl in Environmental Health Perspective. It's a commentary on the concept of translational science that the NIHS came up in 2018. And they tried to articulate what would be a T1. So T1, which is basically what is happening at the bench, is discovery in environmental health science. And usually this is triggered by an observational research. So somebody has seen something, and that has been then communicated, and they start trying to see whether there is some mechanism that actually you can test in an animal model, or you're looking into cells. So I'm putting here a few examples. Asbestos, for example. So these are authors in the 1930s, Meriwether and Price. They start seeing all these cases of people that work in text mills actually coming with respiratory disease, much more than expected. Asking, what is it about the place they're working that could be causing some of this respiratory disease? And that's how the field started looking into asbestos and how asbestos is having an effect. And they'll start looking at mechanisms. And nowadays we understand that asbestos has implications in lung cancer, in development of lung fibrosis, etc. But all that came from the observation. Lead, for example. Lead, some of that is even historical. So historians were talking about in the Roman Empire that emperors used to take in their wine to sweeten, put lead in it. And there was a whole concept of whether all this lead that was added into this wine would be causing some of what we know happened in the Roman Empire, that emperors start becoming mad. Started to create the question of what is happening with lead and neurological. There was an increased death in the 1950s from the London fog because people are getting buried. So this florist started asking, this peak all of a sudden of people dying because the florists are getting more orders. The question is that scientists, and they need to have communication to be able to determine, is there a mechanism that we can understand that could be causing this, or is it just a correlation? And more recently, and the APA, the NIH, and FDA have come up with Tox 21, which is trying to get predictive toxicology. And toxicology is really that area in which, at the bench, trying to understand these doses and these exposures, how they can create or not disease. And the complexity, the whole genome. So you can have certain genes that make you more susceptible to a toxin compared to another person who could be more resistant. Develops lung disease. So, but some people, 30% or 40%, they develop lung disease. So what are the genes associated to that specific exposure? And that's some of the work we've been doing here at the University of Iowa. The T2, T3 is going to be on health policy implications. So you're looking at cross-disciplinary work in environmental health sciences. So for example, T2 would be exposure-response relationship. So now you have you need a team of epidemiologists, toxicologists, biostatisticians, and clinicians to understand how this exposure is related to, for example, the disease that they're seeing in the community. How clinicians start seeing that there were these people exposed to these flavors, that created a whole field that they understood that the flavor caused fibrosis of the airways. And the industry ended up removing that flavor. But that was coming from observation. And that is part of the toxicology component. Not everybody who gets exposed to the flavor and the same amount of flavor will develop lung disease, but there's a dose in which you start developing this. I even had a patient actually that he developed that, and it was after 15 years. And what happened is that he loved popcorn, and he used to do popcorn 3 times a day. But the flavor, such a nice flavor that he would open the bag and just smell it. And doing that, he ended up having the same exposure as somebody who would work in a factory with popcorn. So these are examples of observations that clinicians need to keep in mind and communicate to scientists field, for example, to see how you can try to understand the mechanism of disease. T3 is identification of appropriate interventions to reduce exposure and improve health outcomes. So now, this is the equivalent of a clinical trial. What can I do to try to reduce that exposure that actually could potentially then reduce the number of events that I'm seeing? And it's very much a preventive strategy when you think in the field we're talking about, which is environmental science and exposures. But, for example, the example that we think all the time is going to be related to air pollution. You can think about also in smoking, so trying to have behavioral and strategies to try to reduce that exposure to smoke. T4 is a policy and system change, so it will be implementation of science with a cost-benefit evaluation and decision-making. This now goes outside the realm that I do, and this probably, some of you probably could be involved or they are involved. You start thinking about now the impact of this intervention on, and how would that actually be a cost of benefit, because at the end of the day, a lot of this has to do with regulations, and politicians have to make decisions, and industry has to make decisions. So it's not only about health, but how that intervention also would give you a cost benefit. And you have to articulate this well. And that's one of the challenges when you think about all the issues with climate change and pollution, that there is always a different type of perspectives and interests and incentives. And you need to work in that environment because that's the real world. And the last one is evaluating population health impacts. And this is accountability research. So this is basically, you have the intervention, and now how that intervention has really actually done something. So this is the measurement of the effect of the intervention after their implementation. I really like this example. Pope is very well known in the field on air pollution in particular matter. And Pope and Al estimated in a paper in 2009 that the reduction in air pollution in the U.S. accounted for a 15% overall increase in life expectancy. So it's an actual number that you can put to that. And in addition, the EPA also talked about saving around $2 trillion, because if you're reducing really health-related outcomes, you're having savings on the other side. So if you're improving health, you're reducing all the burden from healthcare, for example. So you have these implications that you need to articulate. And all of a sudden now, politicians who you need to communicate to, the public that you need to communicate to, you can tell them, you know, this is exactly the impact that this has had. And in that paper, I just want to put it because it has a very nice diagram looking at the different T1, T2, T3, T4, and T5, and how they are related to the different types of studies and teams that they need to be involved. I really invite you to read that commentary. It's 6 pages long, and it's really, really insightful. This is the actual environmental health perspective from NIHS. And here trying to talk about translational science, and you have these circles. And there are a lot of different areas in each of them. I don't want to go over everything, but what I want to tell you is that one person cannot really take over this whole process. It's a team, and you need to have everybody in a different aspect of their role in collaborating into translational science. So when you think about translational science, one of the first steps that you need to do is to create that network. That's key. Without that network alone in the bench, you're not going to be able to really take it to the next level. Here we have, for example, this is just an example of the different aspects in this article. Population human observation, so elevated enhanced levels of lead to pilot study in Cincinnati BSERP cohort, and the population human observation. This was half of the Cincinnati girls assessed between 2005 and 2006 had serum concentration about 95th percentile. Then you have an in vitro rodent understanding of PFO stimulates mammary gland development in mice. Then you have also an in situ water identification of probably exposure pathways. You have also another aspect of organizational policy where they partner with city health department to reduce exposure through water filtering. So you have different aspects of a translational component that can go from the in vitro to seeing something in animal model to a population observation to a policy change. And the typical example that I think about a lot, because this is the very basic of what people think about translational science. If you have an observation, a group of population of human, that then you try to take it into an understanding of mechanism. And this would be, for example, an ex vivo mouse model you can imagine. Clinical observation, epidemiological observation, just a small piece of what I want to show you. There are no questions in regards to what I just discussed in the T1, T through T5 or T4, depending on clinical trials and these definitions. And I love that question, that comment, because one of the things that you encounter is that the really impactful interventions and because all of a sudden there is an observation that seems to be relevant in the real world. And I know, for example, one of the areas that there is a lot of still debate is in nanotoxicology. And there was asbestos epidemic. And I think that what I want to bring to the audience today is that you need to probably create these relationships with people who are in the field seeing things, or talking to epidemiologists, or people that are able to measure what is happening out there, that actually there are correlations, but answer questions with regard to potential mechanisms. That makes it extremely powerful when you take that observation and now you take it to the bench. Because you try to articulate in a grant what is the significance. It's very easy to say the significance comes from this epidemiological observation or this clinical observation, as opposed to trying to make a significant argument starting from the bench and not knowing necessarily whether that is reproduced in the real world. So that is a great comment, and I invite people to think in those terms. Any other questions? Okay. So, good time. So, I want to talk about respiratory infections. And when I think about respiratory infections, you think about people who are predisposed to that. So asthma, COPD, people who have cystic fibrosis or pulmonary fibrosis, the extreme ages, young and elderly, the immunosuppressed where the immune system is not able to work, or smokers. And I didn't put here, but also we know that pollution is an increased risk factor to develop respiratory infections. And when you think about respiratory infections, the typical thing clinically that you observe is this. You see what we call a consolidation and infiltrate. Somebody has a cough, shortness of breath, fever. You examine them, their vital signs are a little bit tachycardic, they're a little bit short of breath. And when you do an X-ray, you would see this very clear widening in the lung, which is abnormal. It should look dark. And if you do a CAT scan, which a lot of people nowadays automatically they send to the CT scan of the chest, you see this very dense consolidation that is full actually of pus. That's what it is. And we call these air bronchograms. You can see the airways into this consolidation. And that is diagnostic of pneumonia. Why is respiratory infection so important? Because there's a global burden. And this actually, you can see on all ages, depending on where you live, lower respiratory infections actually have a global burden in different parts of the world. Certain areas of Africa, you can see that actually this is per 100,000. This is the mortality, 100 to 249 per 100,000. In the United States, is around 25 to 49. And there are areas, there are certain hot spots here, for example, in Africa and Ethiopia. But this is all ages. However, if you now look at less than 5 years old, it's even worse. And why is that important? Because I don't know whether you know the Melinda Gates Foundation. They put a lot of their research into the first 5 years of life. And they do that for a rationale. And the rationale is that if you are able to make it the first 5 years of your life, the likelihood you will be able to survive and have a adequate expectancy is much higher. So whatever happens to you in the first 5 years of life matters a lot to that society. And you can see areas here with very high mortality. Areas in India, areas in Africa. When you looked at the United States, we saw that the number was actually not that great, but it's because we have a lot of elderly. So the older you get, the more likely you can develop pneumonia and then die. However, kids, they usually do not die in the United States from pneumonias. While in other parts of the world, you see that kids actually are dying from pneumonia. And it's one of the number one causes of death. Diarrhea has already not gone as first, it's probably third compared to the respiratory infections. So when you think about respiratory infection, we think about the host, the virus, and the bacteria. And my lab has worked into all these mechanisms to try to understand how, for example, pollution can affect the adaptive and the innate immunity as well as the bacteria. And we have done that at the bench, and we have tried to understand how the particles are able to be important nutrients for the bacteria, at the same time impair the ability of the airways to kill bacteria. But what I want to concentrate today is on an observation that this mechanism we looked at in regards to a seasonality effect that we see with respiratory infections. So we're always inhaling particles and bacteria. We're always inhaling. But we don't have respiratory infections all the time. And when you think this is a photomicrograph in the lab of airway epithelia, this is the cilia, which actually is supposed to be beating, and this is a particle, and this is a bacteria. This is Pseudomonas. Together in that environment. So when we're inhaling, we inhale particles, we also inhale bacteria, and our cells have to deal with that. And with the first mechanism of defense that even invertebrates have is these things called antimicrobial peptides. These are small peptides that actually they're positively charged. Bacteria is negative. In bacteria, and this mechanism kills bacteria. Happens to us when we inhale bacteria, we reduce the load by just killing as much as possible, and then the cilia will beat and get them out. And these are antimicrobial peptides are in a liquid, so we have this very thin liquid floating on top of our cells called airway surface liquid. And they have, they're different antimicrobials, but these are the top ones. Human beta defensin, catalycidin, lactoferrin, and lysozyme that Alexander Fleming discovered. The one I'm going to concentrate today is called catalycidin, L37. And but this is what they do. They make a pore, to make a hole into the cell. And why is that important? Because a defect on this mechanism actually translates into disease, in human disease. And here at the University of Iowa, through a pig model, actually, they have discovered that the reason why people with the gene of cystic fibrosis develop respiratory infections is because there is a defect in the ability to kill bacteria to begin with. And that has to do with the pH of the liquid. But the bottom line is that they're not able to deal with the load as well as somebody who does not have cystic fibrosis. So that means that inability to deal with that load translates later on into human disease. And these antimicrobials have been actually associated with people who carry certain bacteria, Staphylococcus aureus, respiratory infections, and also people who have chronic lung disease of the adult, which is COPD. There's been shown to have a role in that. Perfect. So, as I said before, we always inhale this, but you don't see pneumonia, so you don't see respiratory infections throughout the whole year. We know that if you're on service here in the hospital, in the intensive care unit, or in the hospital in the inpatient, in the month of the winter, you're going to see more respiratory infections coming in. So there's a seasonality. Something happens that there are more events and severe happens, for example, in Australia, in the winter time for them. So there is a seasonality event. And here you can see seasonal baseline. These are the reported deaths from pneumonia and influenza and the epidemic threshold. So you see these peaks happening during the winter time. And in Iowa, we know we have different sunshine. So this is, for example, in 2009. Here we can see the amount of sunshine and hours per month. This is during January. Then you get here to June, and you get down here in December. And also we look at not only sunshine, but UV. And why UV index is important, because I'm going to tell you what is one of the things that people have thought about in regards to UV. So the first observation, and this is great because Edgar Hope Simpson, he made incredible observations. He went into all these records. He was the ultimate clinician epidemiologist who would go into the records and look at, influenza cases. And interesting, he's the one who described actually the seasonality of the flu. And it's all because he went into in their hospital, going into their medical record room, getting all the records and spending a year. And he described this in several papers. And here he says something interesting. He says that none of these seasonal characteristics can be explained by the current concept of influenza epidemiology. A new hypothesis recently proposed and recapitulated in appendix offers a possible explanation. The primary agency mediating seasonal control remains unidentified. And then he talks about annually north and then south across the surface of the earth. If it were spreading directly from the sick, the progress should be multi-directional. Following lines of direct effect are the countless adaptations evolved by plants and animals to evade rigorous and seize the opportunity of seasonal changes. So he talks about this seasonality, and he talks about that even having different temperatures and humidity, which people have talked about, does not explain completely this seasonal effect. And he proposes at one point that it could be something related to the sunshine, sun exposure. So what is that important? Because in his paper he shows a monthly percentage of total epidemic month in each zone. So here you can see the latitude. So are you in the north or are you in the south? Then what you have here is the different quarters. And if you're in the tropical area, you see more of a throughout seasonal influenza cases. And then in the north, you see more in the winter time, while in the south, what will be our summertime is their winter time, you see more cases. So he does this, beautiful drawing, and, you have to imagine there are no computers. He's putting all that based on record that he has looked at. So he starts saying, well, there's a seasonality, and the seasonality depends on the latitude you live. So one observation that people have made also is that when you have tuberculosis and there are no treatments, one thing that they would do, they would send you to a sanatorium. And one of the things that they did was have fresh air and close to the sun. And it's interesting because there's been a lot of hypotheses about why cases in the sanatorium actually got better without treatment. And it was more than expected based on the natural history of tuberculosis. So this antimicrobial I mentioned to you, L37, actually gets activated by vitamin D. And the vitamin D in us gets active, so we have a storage by having UV exposure. So you have UV exposure, you have the 7-dehydrocholesterol, it goes through all these cycles into calcitriol, and then 125 is the active form. And that active form is important because it goes into the cell, it goes into this vitamin D response element and transcribes this antimicrobial peptide. And what is interesting is that this is something that has been conserved in evolution by mammalians, but is not present in rodents. So you cannot study this in mouse. And mouse, they are nocturnal animals. They're not exposed to the sun. So when you think about some of these mechanisms, you have to take into consideration what is the right animal model. And it's interesting that evolution has some selection depending on what are your habits. So the hypothesis we had, knowing that there's a seasonality effect, that vitamin D actually seems to be important to transcribe this, and understanding that sun exposure will affect on your vitamin D levels, we hypothesized that airway antimicrobial activity will explain in part seasonality in respiratory infections. So airway antimicrobial activity will show seasonality. That's what we thought. You would be able to see a seasonal effect in the ability of your airways to kill. But how do you do that? So we were lucky enough to be able to collaborate with a colleague who actually had this trial called the lung response to respiratory pathogens. So these people in Iowa were recruited, and they got bronchoscopies. So they got scopes. And they got it throughout, they got a first scope, and then they got a second scope 90 days later. But everybody who come in to get a scope, they go also vitamin D level checked. And they were randomized to get vitamin D, depending on whether you were placebo or what they collected. You have liquid from people who come throughout. They get a scope before, they get a scope after, and some people get vitamin D and other people don't get vitamin D. So we had a unique opportunity to ask a question in this population about, is there a difference in the ability to kill bacteria or not at baseline? And would you have a different response if I give you vitamin D? And to do that, what we do is we do a simple assay, which basically we have is cells that they have this liquid, but also I can take the liquid from my patients. I could put it into these wells, and or the subjects, and then there is this bacteria that emits light. So when I throw the bacteria on the liquid, if the liquid can kill light you have tells you how much live bacteria is there. And because this kills immediately, by putting this bacteria in there, you're able to measure this within a couple of minutes, because that's how fast these antimicrobials kill. So we had these bronchoscopies with the airway surface liquid collection pre. This is a collaborator, Alicia Gerke, who's part of the center too. And these people, as I said, they go, they get a scope, and then what we did is we take this fluid and we put it in the well, we challenge the fluid with bacteria, and we see how much light is there. So the first thing is that this is the amount of live bacteria. So at baseline, people coming is by 3 months. And what you find is that there is more live bacteria when I challenge that fluid in the people who came in the winter time than the people who came in the summertime. So you could say, well, it could be different. It could be that the people in the summertime, they are able to kill better than the people who are in the winter time. But there was an interesting signal that actually there was a difference in the ability to kill bacteria if you came in the winter time versus the summertime. So the ASL antimicrobial activity is lower during the winter compared to the summer. As I said before, there's a vitamin D metabolism, and you need to get 125, which is the active form. So what you can see is what is the vitamin D levels on these people. And if we check the vitamin D levels in any of us at different times of the year, you will our vitamin D levels are lower. It's because of the UV exposure and the sun. And what happened is that the amount of vitamin D in the people in the winter time that has less killing was lower compared to the summertime. And when you group them by winter, spring versus summer and fall, there's a difference in their vitamin D. And this number 20 is what you would say anybody below 20 is clearly deficient. That has effect even on your bones. There's a bunch of people down here in the winter time that they're below 20 compared to only a couple here. Ultraviolet index in Iowa, actually, in Iowa City. So we had that data. And you can see that the UV is much lower in the winter time than the summertime. And we measure the amount of vitamin D, active vitamin D, between winter, spring versus summer and fall. And not only the total amount, but the active form was also higher in the summer compared to the winter time. So vitamin D levels are lower during the winter time. The other thing I mentioned to you is that this vitamin D response element will then increase the amount of production of this antimicrobial peptide. And the specific one is a catalycidin that I mentioned to you before. So we cannot really measure the antimicrobial peptide because the amount of liquid is very, very small. So one thing that you can do is you can put an antibody against that antimicrobial. You bind it and you challenge that liquid with the bacteria. If that antimicrobial is not available, it cannot kill. So by treating the liquid with the antimicrobial peptide, we found that that difference was not present anymore. And we have done that even with IgG as a non-specific antibody, and we find that actually still the difference exists. So this was indirect evidence that actually treating against one of the antimicrobials, that seasonal effect disappeared. So blocking L37 was sufficient to abrogate the ASL seasonality difference. But I said to you that actually these people got vitamin D. So you would argue that people who are deficient of vitamin D and their antimicrobial activity was lower, if I give them vitamin D, I should be able to mount an increase in the antimicrobial activity. While the people who were killing already fine, not necessarily are going to kill more. So when you look at the vitamin D levels, this is vitamin D levels in the winter, spring. When they received vitamin D, there was an increase. The placebo, there was no difference. In the summer, the vitamin D actually levels were not increased, neither the placebo. And the reason why you don't increase vitamin D levels is that if they're good by giving you more vitamin D, you're not going to have more vitamin D. It's going to just be a feedback mechanism that actually inhibits the amount of activation of vitamin D. And when you look at the antimicrobial activity, the people that actually got vitamin D during winter time, they kill more compared to the people who were getting placebo. In the summer, you don't see any difference because they're already killing more. So the bottom line of this is that the vitamin D that was given to these subjects who were in the winter time and their ability to kill bacteria in the airways was reduced had a positive effect in increasing the killing. While the people who were already established with adequate levels of vitamin D and adequate ability to kill bacteria, they were not able to see any difference. So vitamin D supply during winter increased vitamin D levels and ASL antimicrobial activity, while during the summertime did not have any effect on the serum concentrations or the killing ability. So the conclusion of this experiment, which was taking advantage of an observation has been epidemiologically, which is seasonality. Seasonality have seen that you have issues of increased respiratory infections. We see that this seasonality has not been explained. We do not know why people have more respiratory infections during the winter time compared to the summertime. And this epidemiological observation in regards to an environmental situation that we do not identify what it could be, we hypothesize based on a clinical trial that already was there to take advantage of and those samples to try to understand whether there is a mechanism or not. And what we learn is that actually the antimicrobial activity of the airways seems to follow a seasonal pattern driven by variations in serum vitamin D levels that affect the antimicrobial peptides in the airway, specifically the catalycidin, which is L37. And the vitamin D supplementation increased vitamin D serum levels and ASL antimicrobial activity during winter and not summer. And the reason why is that important because there's been a lot of clinical trials that people give vitamin D for the whole year, don't see an effect. And it could be that you don't see an effect because you're having also the situation that people are getting therapy when there's no effective since it's summertime. But if you just direct therapy during the time that they're more susceptible, it could be that actually you could potentially reduce that risk. So we want to continue following this study to validate it in a prospective manner because this is retrospective from samples that were already been selected. But the example I wanted to give you here was really, and this is the team of people that I work with, and they're from different walks of life, computer science, chemistry, environmental health, radiology, ID, and computer science, chemistry. So all these people in my lab, we work together to try to answer some mechanistic questions. But these mechanistic questions are being looked at from the perspective of an observation that is happening either clinically or is occurring epidemiologically. And then we're able to say, oh, is there a mechanism? And if there is a mechanism, can we now create a clinical trial or have an intervention, which will be that T2 to T3 intervention in which you see whether you're reducing the risk. So you can imagine that one intervention could be vitamin D. The other intervention could be actually UV exposure. There are issues with that because you have increased risk of skin cancer. So there you have to take into consideration all that. But actually, you could have a design to prove that this observation actually is translated into a reduced risk of developing respiratory infections. And all this is, we think, related to an environmental issue, which is what? Sun exposure. People are not necessarily having the same sun exposure during wintertime, during summertime. You can imagine the same thing could happen in people who live in places with high levels of air pollution. So not only the pollutant can actually affect it, but also we know that pollution can block sun. And that has effects in the ability for you to be able to be exposed to UV, to be able to convert into vitamin D and have increased antimicrobial activity. So this was just an example of how you're trying to translate these observations at the bench, and now what we want to take this from the bench into the bedside and see whether we are able to determine the effectiveness of that. So with that, I would like to stop and then answer any questions you have about this talk on translational science and environmental research. And thank you. Yes. Yeah, so there are airway epithelia, produce that. Macrophages produce that. They're also in the skin. So skin also can produce that. So they're present specifically in epithelial cells. That's where we see them a lot. GI tract, I believe, also they have. So it's a very ubiquitous antimicrobial that has this ability to kill.